most of the information is available on Spectrum. We have a few extra documents that I think is helpful for you. On Spectrum, I have the DIFCO manual there. It's a catalog of all the medias that they produce. It's useful because for every media that they list in the catalog, it tells you the principle, why it works, how it works. So it tells you what are the components that selects the bacteria, what are the components that differentiate the bacteria. Okay, so this will increase your theoretical knowledge. It also has recommended positive and negative controls. If you are working in an ISO lab, you need to know how to quality control your media, to know whether your media is still working. Okay, so these are important information. You understand? Okay. So so this is useful. If you download it and keep it. If you are working as a microbiologist, you will find that it is useful. Okay. Okay. So this is the learning outcomes that we've gone through. So as I've mentioned, let me repeat. We only do the important bacteria, okay? So these are some of the course contents. These are the selected references. So as a bacteriologist, you have several alternatives. You can either go into the clinical line, medical, or you can go into industrial. For industry, you have pharmaceutical, you have chemical, especially any chemical that contacts with the human skin, human application, which means detergent, soaps, everything has to have a microbiologist inside. You need to do microbiology testing. So for industry, food, pharmaceutical, and even and detergents and all that. So the job of a microbiologist is, first you have to be able to follow instructions. All established labs already have SOPs, okay, standard operation procedures, which means you don't have to reinvent the wheel. You only need to, sometimes if you are working in a private lab, a client comes and say, I want to identify something new. Then you have to use your scientific knowledge and expertise to look for information on how best to isolate and identify the bacteria. Okay? But most of the time, you just follow the standard SOPs. Okay? And within different industries, the types of bacteria they are looking for are all very standard. Okay? Nothing surprising. That's why you keep seeing the same bacteria again and again. Okay? So if you're in the clinical line, if you're in the hospitals, you keep seeing the same bacteria emerging again and again. So to identify bacteria, first you have to isolate, right? First you have to isolate. So this is essential. Exercise in food and healthcare. How many ways are there to isolate a bacteria? You have broth culture, you have solid media culture, right? You have enrichment culture. So lots of different ways to isolate the bacteria, and that is only talking about your media. But you also have to be concerned about your environmental conditions. Do you incubate at 37 degrees Celsius? Do you incubate at 30? Do you incubate at 4? Okay. After isolation and culture of bacteria, bacterial isolates should be purified. So you've done this, right, last year? dilution streaking. How many times should you purify? Minimum? Best practice 3 times. Best practice. If you have luxury of time. Because you know that if you do the dilution streaking, then you have to wait for it to grow before you can do another one. Okay? In private labs, you do not have that luxury. Okay? They want the results immediately or as soon as possible. But if you are doing research, then you can do it because you need to make sure that it's pure before you continue. Okay? So, bacterial identification is for practical purposes, which means you use it in food industry, water treatment, clinical laboratory. Coliform, that's an interesting word. It's a group of bacteria. A group of bacteria, which means the basis is you use media that has lactose, you culture them, if it grows, so they are lactose fermenters. And anything that grows in McConkey, for example, you all know McConkey? Okay, so anything that grows there, they are considered coliform. Pinkish, reddish colonies. Because they are lactose fermenters. Okay? So that's the group of bacteria. So on the basis that these are the ones that can cause gastrointestinal diseases. Okay? So Escherichia coli, very important bacteria in the food industry. What else? Any other bacteria they all know? Pardon? Okay. Right, Vibrio cholerae. What disease does it cause? Cholera, yeah. So remember, since we cannot write italicized, we have to underline it. Okay? What other bacteria important in the food industry? Listeria. Oh, you all learned this already. The main one is Listeria monocytogenes. Okay? But this is gram-positive. Okay? This is usually, it's also important, but only for selected types of food. So we also have Salmonella, we have Salmonella typhi, Typhimurium and all that. You have Shigella. You can have dysentery, Clostridium botulinum. Gram-positive also. Okay, you have lots of bacteria. So these are gram-negative, 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 gram-negative. All this will be covered in the course. Okay? Then water treatment. Water treatment, when you talk about water treatment, the term coliform will come up. Then you also know about fecal coliform. Then you have E. coli. So for water treatment, coliform is a group of lactose fermenters. Okay? Anything that grows on media that has lactose and can ferment it, those are coliform. Fecal coliform are lactose fermenters that grow well at 44.5 degrees Celsius. Okay. So usually the procedure is to isolate for coliform first, and then further incubate at 44 to see which one grows. The ones that grows are fecal coliform. The ones that don't are just coliform. Okay? So these are groups of bacteria. But now they've refined the method to focus mainly on E. coli. So these are bacteria, groups of bacteria and bacteria that are important in water treatment because they are considered fecal indicator bacteria, which means if you can detect this bacteria in your water supply, there is fecal pollution, okay? A recent fecal pollution in that water source. Okay, so that is why these are not often, it's mandatory. You test in water treatment systems, okay? Then, if you are interested in clinical laboratory, so you have about one full semester, right, in internship. During your internship, if you're interested to work in hospitals or clinical laboratory, apply for hospital internships, because in hospital they will rotate you through all the different units. You go through hematology, parasitology, biochemistry, and microbiology. So that is better training for you. Okay? That's my advice. So if you're interested in medical microbiology, you go for your internship in hospitals. Okay? The government hospitals have very good training in the past. I'm not sure, but just ask your seniors first before you choose which hospitals you go to, because they will know the most recent, whether they still implement good training. Okay? Then, because it is for practical purpose, that's why bacterial identification is restricted to a few common and important bacteria. It's not like research. You know, when we do research, we want to know, usually like the whole community profile. We want to know all the types of bacteria. And now with NGS, high throughput sequencing, next generation sequencing, we are able to see lots and lots of bacteria types in your sample. So if you're looking at all, you need to go for NGS. Okay? But if you're looking for selected, then you can still use culture approach. That means culturing is not dead science. You still need it, because whatever you get from NGS, you can't really apply it immediately. But if you can get the isolate out, immediately you can work with the culture. Okay? So it's faster in a sense that way. If you want to look for rare bacteria, what should you do? The first thing you should do is you shouldn't do what others have been doing. If you do what others have been doing, you will get the same results as they. Understand? So it has to be something different. A different media, different conditions, then you'll have different bacteria growing. The standard identification is now we have 16S rRNA gene sequence. Okay? So the gene sequence is about 1,500 base pair. So that gives enough information for you to identify the bacteria to species level. So in identification, In bacterial identification, imagine if you're in the lab, you're doing identification, the first thing you usually do will be, once you've isolated them, you have to do your staining, right? Yes? Staining. You need to stain them, right? From your microscopy, you can see some of the morphology. So when you do characterization of the cell wall, what does that mean? Characterization of the cell wall means it's either a gram positive or gram negative, right? Or, for gram, there's also a gram variable. Gram variable. When you're working with common bacteria, usually the results are very clear-cut. It's only when you're working with environmental bacteria, some of the bacteria gives gram variable results. Okay? So you know that gram staining is based on cell wall. That means your culture should be fresh, right? If your culture is not fresh, it doesn't hold the cell wall doesn't hold the stain that well, then you'll have results that are not so consistent. We also look for acid fast within that cell wall itself, right? Because some bacteria has mycolic acids that are able to be tested for acid fast stains. So this phenotypic test, so things that you can see that are of taxonomic value, just the cell wall differences, gram staining, acid fast stain. What about bacteria that does not have any cell wall? Mycoplasma. This is no cell wall. So if there's no cell wall, what do they stain? There's no peptidoglycan to hold the purple. That means your counter stain is what you see. Okay? So you also have other morphology, capsule. Capsule usually is look through what? You've done this, right? It's through negative staining. Which means you stain the background and you want to see the capsule surrounding the cell. It's important because most pathogenic bacteria has capsule and capsule. Okay, that's how the bacteria defends itself. Okay. Motility. How do you check for motility? Step agar. You all know step agar? It's test tube, soft agar. You stab it and see it grows. If it grows around this area, it's not motile. If it's motile, it will be cloudy. Okay? Because it spreads. How much is soft? The normal agar concentration in your agar plate, how much is that? Usually, if it's pre-mixed, then you don't have to worry. But sometimes you have to add your agar yourself. So you can choose between this range. But soft agar is about 0.4. So that will give you soft agar for you to stab. Stabbing cannot be too near the bottom. Okay? Because then if it spreads like this way, your results will not be accurate. This is probably the most reliable way to getting motility results. a quick and easy way would be to look under the hemocytometer itself, see whether they are swimming. Okay. Of course, you can test for flagella, but it is not easy to stain for it. Pigmentation, some bacteria produces color, but only at a colony level can you see. Because a single bacteria is too small, whatever color it has, you cannot see. Okay, it has to be from the colony. The thing that you have to remember is the shape of the bacteria, the colony. The shape of the colony depends on the media and the growth conditions. Okay, so make sure it's the standard one that you're using so that you can compare. Okay, remember that. This one depends on the type of media and also the incubation conditions. Then also spore formation. You have your endospore. Endospore usually more for gram-positive bacteria. In the food industry, the ones that are of frequent appearance is your bacillus and your clostridium. What's the difference between a bacillus and a clostridium? So you remember that the location of the endospore can also be important in taxonomy, whether it's at central or terminal or subterminal. Clostridium endospores are larger than the diameter of the cell. That's why there will be a bulging. Bacillus endospores are smaller or similar to the diameter of the cell, so you won't see any bulging. So this difference actually helps in telling you whether you are working with a bacillus or clostridium or something else, because not only clostridium, you also have anoxibacillus that can do this. But in the food industry, medical, these are the main bacteria that will cause problems. Okay. Another difference, this can be facultative, this is anaerobic, obligate. Okay, obligate anaerobe. So Clostridium is an obligate anaerobe. Bacillus is a facultative or aerobic. Biochemical tests, including nutritional requirements and acid production. So all these tests checks whether your bacteria has that enzyme, okay, is able to ferment a certain sugar, okay, and from there you are able to identify the bacteria.